Cancer is a disease in which the body's cells grow abnormally and uncontrollably, with potential to spread to various parts of the body. It is also among the leading causes of death worldwide. Researchers and aspiring scientists, like myself, are still trying to figure out how this mysterious disease works. Hi, my name is Shannon, and I am an incoming sophomore in the College of Language Science at the Arts here at the University of Michigan. I'm trying to understand what is really going on in a type of cancer called T-cell acute lymphoblastic leukemia, or TALL. Right now, I'm helping my lab study an oncogene, or a gene that has potential to cause cancer, that is activated in this type of leukemia, and what role the various proteins that interact with this oncogene play in the grand scheme of things. But before we can fully understand what happens on the molecular scale in a disease like TAL, we first have to understand how does our immune system, specifically our white blood cells, protect us from pathogens, and what happens when they become cancerous. Our immune system has two parts, the first being the innate immune system, in which we are born with and attacks all general threats, and the adaptive immune system, which our bodies develop over time. The adaptive immune system, otherwise known as adaptive immunity, relies primarily on T cells and B cells to target and eradicate threats. These immune cells are formed from a specific type of stem cell in the bone marrow. Now, let's explore these two types of immune cells. After leaving the bone marrow, B cells move through the body via the lymphatic system. B cells mature as they encounter and engage with antigens. An antigen is something that is present on the outside of a pathogen that triggers an immune response in our bodies. Think of it sort of like an ID tag that identifies the pathogen as such. But you may be wondering, how do these antigens actually trigger an immune response? Turns out, of the billions of B cells that circulate the body, they each carry a receptor on their surface that is specific to a particular antigen. When a B cell that has not yet fully matured encounters an antigen that fits its receptor, the B cell starts dividing into either a memory B cell or an effector B cell, also called plasma cell, in a process called differentiation. Think of this kind of like a puzzle. The antigen is one piece and the antigen receptor is another. The original B cell is now called the quote-unquote parent cell. Both the memory B cells and the effector B cells have the same receptor as the parent cell, but the difference between the two is that the memory B cells carry the antibody on the cell surface, while the effector B cells are able to release the antibodies. These free-floating antibodies work by locking onto pathogens in the body and marking it for destruction by other immune cells. Premature T cells, on the other hand, migrate to the thymus after they are formed in the bone marrow. Once matured, we are left with three types of mature T cells: helper T cells, cytotoxic T cells, and T regulatory cells. Helper T cells help to activate cytotoxic T cells, B cells, and other immune cells. Cytotoxic T cells help to get rid of pathogens and infected host cells. T regulatory cells help the body determine which molecules are self and which are foreign. This is important because it prevents the body from accidentally attacking itself. Today we'll be focusing on helper T cells and cytotoxic T cells. Think of them kind of like an elite unit. The cytotoxic T cells do the destroying and the helper T cells coordinate the whole thing. They circulate the body and look for antigens to differentiate between normal cells in the body and infected or intruder cells. Healthy cells present self-antigens, while cells infected by a virus will present pieces of virus antigens on its surface. Like B cells, each T cell carries a unique T cell receptor that can only fit one kind of antigen, much like how a key pairs with only one lock. So when a cytotoxic T cell finds a matching antigen, it releases proteins and toxins, creating pores and ultimately destroying the foreign body. Helper T cells, if you recall, are kind of like managers and directors that help to coordinate attacks. They give instructions to other immune cells by sending out chemical messages. The chemical message alerts the cytotoxic T cells when there are pathogens. After they destroy the pathogens, the message tells the cytotoxic T cells to keep making copies of itself, kind of like creating a unit of soldiers that specialize in destroying that specific target. Helper T cells also stimulate B cells into making more antibodies. But what happens when these immune cells become cancerous, like in leukemia? Normally, the bone marrow makes blood stem cells that mature into either myeloid stem cells or lymphoid stem cells. Lymphoid stem cells eventually become one of three types of white blood cells: two of which we talked about earlier: B cells, T cells, and natural killer cells. However, in the case of leukemia, there is an overproduction of immature white blood cells, which do not function normally and are not able to fight infection very well. They could eventually multiply to the point of crowding out healthy white and red blood cells and platelets. This could result in increased infection and anemia, among other symptoms. Today, we learned a little bit about how our immune systems work to keep us healthy. There is still so much work to be done, not only to better understand immune cells and what happens when they become cancerous, but also to develop therapies for people with leukemia. My goal is to ultimately add to our current understanding of this field and help to improve the lives of people with leukemia through research.